Hi, welcome back to Bio 131. We're starting chapter 28 on eukaryotes and eukaryote diversity. And so I've taken this chapter, I'm divided it up into 4 mini lectures. The first, if you've had 130 or Bio 102 recently, you may be able to skip. I'm basically just going to go over what is a eukaryote cell, focusing mostly on the organelles, and very importantly, the ability to do phagocytosis and the endomembrane system with the nucleus. So if you're familiar with all of those terms, you can skip on to the second lecture on the origins of the eukaryotes. And so what I'd like to do is go over and describe, just as I mentioned in this little mini lecture, what is a eukaryote cell, what it looks like structurally. This is going to refer to things in diagrams from earlier in the book, but you only really need to know the overall view. You don't need to know what each organelle does and how they transfer and all the stuff that would be covered at cell biology in 130. So you don't need to know it in the whole detail that is in the earlier chapters. Okay, so let's get going here. And so we're talking about the origins of eukaryote diversity. Outside there. And just today we're going to be going over characteristics of eukaryotes. And so what is in yellow here is today's lecture. And then I will break up and do another lecture on the origins, how it forms the nucleus endomembrane system, mitochondria, chloroplasts. And we'll continue on from there to other lectures on serial endosymbiosis, things you don't even know about yet. And then mitosis and meiosis, again, something that if you've had 130, you may know something about. And that'll be another little mini lecture at the end. And so what are prokaryotes? And so we just finished talking about them, and we know that there are 2 basic classes, or domains actually, big groups. And those are the bacteria that more people are familiar with, and the archaea. In terms of their structure, if you remember, they're very simple. Right? In terms of eukaryotes, what we're going to have is basically everything you can see with your naked eye is a eukaryote. And so, of course, it includes the animal kingdom, green algae and plants, and red algae making a big domain or supergroup or kingdom of the Archaeplastida, but all the plants. The fungi, including yeasts that we'll talk about, they're all a kingdom. And then a whole bunch of other smaller organisms, typically with some exceptions like seaweeds and the kelps and the brown algae. But what we'll be going over in the Protist lab is you're going to be seeing lots of diversity of other types of organisms like red algae, brown algae, diatoms, amoebas, euglenas, paramecia, apicomplexans, malaria, so there's internal parasites, water moles, and dinoflagellates, right? So there's a lot of diversity of different things. And so what I'd like to do is review quickly the difference between a prokaryote and a eukaryote. And so if you remember, I drew a prokaryote. We're just going to have a cell wall. Inside, there's a cell membrane. It does have DNA, but as a single chromosome. Not in a nucleus. And then we have the rigid flagella to the outside. And we have these little, oh, this would be green, organelles called ribosomes. Okay, so that was pretty much our prokaryote, right? When we get to the eukaryote, and we'll see a nice diagram in a minute, some have cell walls to the outside. Plants do, fungi do, dinoflagellates, diatoms. We have a lot of diversity, and we have diversity of compounds that make up the cell wall. Inside it, we have the cell membrane. And some have a cell membrane as the outer covering, like an amoeba. Others have the cell membrane inside the cell wall. And what's going to make this difference is to the outside we have what we call the plasma cell membrane. But inside there's going to be other parts of the cell that are also made up of membrane. And these will include the surrounding of the nucleus, they're going to have the endomembrane system. They will have an organelle called a mitochondria. And I'll do one as a plant. Usually ones that are photosynthetic have cell walls. We have chloroplast. I'll make this one green, but we'll see red and yellow and brown ones as well. And they still have ribosomes. They can have an extension to the outside called a flagella. We'll make that orange. But this flagella, notice I'm using the orange like the membrane, is surrounded by membranes and extension of the cytoplasm. So it's very different from the flagella that we saw in the prokaryotes. Okay, so that's my messy diagram. This is what it actually looks like in the nice textbook. So this would be a cell. In 130, you kind of learn just basically plant cells and animal cells. So this would be more similar to an animal cell. But it could be something similar to like an amoeba or a variety of things that we'll see in lab that aren't in the animal kingdom. But if you notice in here, they have lots of organelles that are listed, right? And we're not going to go over all the functions. That's more 130. But I want to focus in on just a few. And that has to be the nucleus. This is in purple. Then in blue is the ER. Then in green is the Golgi. And then to the outside is the plasma membrane. And that's in orange. All of those go together and make what's called the endomembrane system. And what I didn't draw on my little diagram, and I'll mention here, is inside the nucleus, you do have DNA, but as multiple separate chromosomes. A little bit different from the single chromosome that we saw in the prokaryotes. And plasmids are very rare. Plasmids basically don't exist. Very few, maybe in yeast is the one example I know of in eukaryotes, but plasmids don't exist very much. In eukaryotes, so very different from prokaryotes. Okay, we can see the flagella going to the outside, and here's a picture of a plant cell. You can see this rigid cell wall made up of cellulose. In the animal, you can see the mitochondria. In the plant, they have mitochondria, and they also have chloroplasts, and they also have something called a central vacuole, which we'll get to when we get to water transport. It's very important. But notice they still have, in plants, just like animals, they have the endomembrane system. You have the purple membrane around the nucleus, and if you look very carefully, it's actually physically connected to and part of the ER membrane coming off of the nucleus. It sends little vesicles over to the Golgi, and the Golgi send vesicles over to the cell membrane. So inside the cell wall, you have the cell membrane, just like in an animal cell. And so we're going to see that there's a flow from the nucleus to the ER to the Golgi to the cell membrane to the outside that connects those different types of membranes, and that is the endomembrane system. Huge diversity in terms of the type. So one of the ones we'll see in lab is called Euglena. It's green, does photosynthesis, and so you might think very quickly that's related to plants, and it's not. It's its own, what we'll call supergroups, which we'll get to in one of the little later mini lectures. And so if you notice a couple of things different about it, it does not have a cell wall, where plants do have a cell wall. It has something called a pellicle, or to the outside, giving it some of these protein bands that give it support. But it does have chloroplasts. It also has a flagella, and it swims around, where plants typically don't swim around. And so it's just showing you some of the diversity that we have in the eukaryotes, and we're going to cover a lot of that in lab. See lots of different types. And so Euglena has a couple things just to point out. It has a long flagella, but notice the flagella here has little extensions on the side. So this is a brushy or a tinsel type flagella. It has a little area here that's called the eye spot, and it's a single cell. It doesn't have an eye or a brain, but it uses it to detect light, and there's a swollen area on the flagella, and if it's swimming towards the light, it gets that signal and it'll keep going. And if it's not, it kind of goes around in circles. Kind of similar to what we saw with the bacteria in the taxus, and this would be something similar to that. There's a second shorter flagella that's in the side there. It has contractile vacuoles. This is a membrane-bound organelle that helps pump water out to maintain its osmolarity balance with the cell, osmotic balance. And of course, it has nucleus. And so features of eukaryotes that we want to focus in on is, first of all, they have a nucleus. They all have a nucleus. They all that exist have mitochondria. They all have that. And then some have chloroplasts, which are one type of an organelle we call plastids. Right? They all have an endomembrane system. They all have a cytoskeleton that helps pull and move things. I'm going to show you a quick little video at the end of this mini lecture on phagocytosis, and that's part of what allows it to do phagocytosis. They have a flexible flagella. They have multiple chromosomes. And then we're going to get to some stages later on that we'll talk about in the mini lecture, and we'll contrast diploid and haploid and talk about the difference between two types of cell division, mitosis and meiosis. And if you remember, in the prokaryotes, they have one type called binary fission. Okay, so let's look at some of the features that make eukaryotes different. And so as I mentioned earlier, the prokaryote had a solid flagella, where the eukaryote has a very flexible flagella. So it moves like a snake. It would swim in the water like a snake. So it's powered along the whole length of the flagella as it's moving along. And there's two types. If there's just one to 4, it's flagella and they're long. If there's many of them and they're short, those are what we call cilia. A flagella is for swimming. It propels the cell. Cilia can do two things. They either can do swimming, or they can move things along the surface of a cell. So right here, here's a paramecia, and it would actually have a food particle and it would move it toward what we call the oral groove, where it could take it in by phagocytosis and feed. What they have is this internal structure of microtubules, and if you've had 130, they're arranged with 9 going around the side and 2 in the middle, so it's called 9 plus 2 microtubules. What you have to remember for us is that the flagella in the eukaryote is very different from flagella and cilia in the eukaryotes are very, very different from the solid flagella that we saw in the prokaryotes. Here's more another picture of its structure. So you notice these are from chapter 6. So you can go back and look at the diagrams in chapter 6. You don't need to know all the details. We're not taking up chapter 6. You just want to be able to tell a eukaryote cell from a prokaryote cell. Right? A couple things about eukaryotes is that they are larger and they have more DNA per cell. So an individual cell can do more things than an individual bacteria can do. Right? That being said, again, if you remember, the bacteria as a domain have a lot more enzymes and chemistry than the eukaryotes as a domain. But each individual cell, a eukaryote can do more. Again, I want to go over and talk about what is the endomembrane system. And so in a cell, we'll have membrane that grows around the nucleus. And if you notice right here, they're showing you see those lines, how they connect? Right? So you could see that that space between, there's actually a double membrane around the nucleus, and that space is contiguous with that space that forms the inside of the what's called the ER, endoplasmic reticulum. And then the ER is attached or sends out little vesicles. Each of these little membrane-bound balloons inside the cell carrying something is called a vesicle. And so the ER makes vesicles that go to the Golgi, and then the Golgi process. Basically, the ER is a big manufacturing area in the cell. The Golgi kind of assembles and labels things so they get to the right parts of the cell. and then some of these vesicles can fuse with the plasma membrane to the outside. And so when we're talking about the origin of eukaryotes, one of the things we're going to have to talk about is where does the nucleus, because we talk about eukaryotes having membrane-bound organelles, but the nucleus is just one of them, right? The organelles that have membranes are the nucleus, the ER, the Golgi, and then surrounding the mitochondria and the chloroplasts are also membranes, right? And they're actually made up of membranes. So those are our membrane-bound organelles. And so we want to know about the endomembrane system because we'll talk about it for its origin. The second thing that eukaryote cells can do that we do not see in prokaryotes is inside the cell there's these long tubules of protein. One's called actin, kind of similar to what we see in muscles. And they can actually pull along each other. So they can actually pull and move things throughout the cell. But they also can take the cell and pull on the membrane so the cell will divot in. So if this is a food particle, let's say that's a bacteria. Because remember, your amoeba, your eukaryotes are much larger than bacteria. As it's coming along, and I'll give its nucleus there, it can actually surround that bacteria and then take it inside the cell. That's by phagocytosis. Notice that the outer membrane here pinches in and surrounds it and becomes the inner membrane of the food vesicle. So that's showing you how these membranes can move. I've shown you in the endomembrane system from the inside to the outside. Here's one going from the outside to the inside. And so here's a picture showing you an amoeba actually eating another eukaryote, in this case a Paramecia. And I'll end this with a little video clip that is from your textbook. And hopefully if we can come onto campus for lab, one of the things we'll definitely be showing you live is showing you this amoeba.
Amoeba proteus is a large unicellular organism that moves over its substrate by sending extensions of its cytoplasm, called pseudopodia.
Amoeba proteus is a large unicellular organism that moves over its substrate by sending extensions of its cytoplasm, called pseudopodia, in various directions. The pseudopodia can surround and engulf food by phagocytosis.
The ectoplasm contains
The
large
leading edge of
amounts
the
of
amoeba
actin,
is a
the
clear zone
contractile
that seems
protein
devoid of
common in animal
cytoplasmic
muscle
organelles.
cells.
This is
The
called
rest
the
of the
ectoplasm.
cytoplasm is called the
The
endoplasm.
ectoplasm contains large amounts of actin, the contractile
And is
protein
then
common
converted
in
to
animal muscle
ectoplasm
cells.
just below the
The
plasma
rest of the
membrane.
cytoplasm is
As it
called the
flows backward
endoplasm.
around the outside of the cell,
During
and
amoeboid
is
movement,
converted back to
endoplasm
endoplasm,
flows forward
a
to
cycle
pull
that
an extending
continues while the
pseudopodia
amoeba moves.
and is then converted to
Credit,
ectoplasm just
Henry
below the plasma
Mainwaring,
membrane.
Western Carolina University.
Ectoplasm flows backward around the outside of the cell and is then converted back to endoplasm. A cycle that
Okay, that's the
continues
end
while
of this
the
little introduction to the eukaryotes.
Henry Mainwaring,
Hopefully, as I
Western
mentioned, we'll
Carolina
see a
University.
live moving amoeba in lab under your microscope so you can see this organism. From here, the next part is going to be talking about the origins of eukaryotes, and we'll do that in my next little mini lecture, part 2.